TARC is a unique structure insofar as the state of Texas has really emphasized that learning more about Alzheimer's disease, diagnosing it, and treating it for the citizens of Texas is an important goal, to bring together the multiple institutions in Texas who are doing work on Alzheimer's disease.
TARC involves all of the top 10 medical schools in the state, and that encourages collaboration across institutions as well. So one of the ideas of TARC was to help us get out of our academic silos, if you will, and find out what other investigators in our own state are doing.
Research is a team science or team sport. No one individual can do it all. We partner up with other individuals with different skill sets and specialties and look at things from different angles.
Having everyone at the table at the same time allows you to see things from angles that you might not otherwise have seen. And it's helpful to think not only about the moment that you're working in or the specific thing you're focused on, but how it fits into the bigger picture.
The kinds of problems that we need to solve are very, very complex. They're beyond the scope quite often of one laboratory or one investigator, and sharing resources, sharing ideas and methods is really the way to come up with the most effective answers to some of these really challenging problems.
This kind of work is not like a terminal project that has a defined start and a defined stop, because one discovery rolls out and that discovery opens the door to something very useful.
It's always amazing to me how people can see the same set of data and find different things interesting or worthy of study, and that's great because nobody has all the answers.
The collaboration aspect allows us to do more than we can do at our site alone. Together we can accomplish much more in more diverse populations with a bigger impact.
It takes a village, it takes a state, it takes a country to fight something that is so challenging and has so many ramifications.
It's a formidable force in research if you're interested in the dementias. So TARC is just going to continue to grow, not only in the research that it produces, but also in its understanding of what needs to be at the forefront of research and what needs to be the priorities of researchers in this field of Alzheimer's disease and related dementias.
And together, we're stronger and better than any one of us alone.
Welcome to the 2024 TARC Scientific Symposium. Please welcome the Scientific Director of the Texas Alzheimer's Research and Care Consortium, Dr. Monroe Cullum. Dr. Cullum is a professor of psychiatry, neurology, and neurological surgery, and serves as the Vice Chair and the Chief of the Division of Psychology and the Department of Psychiatry at UT Southwestern Medical Center. He holds the Pamela Blumenthal Distinguished Professorship in Clinical Psychology and is the Senior Neuropsychologist in the O'Donnell Brain Institute.
I was waiting for the VOG, the voice of God, so to speak. So now we all know how God sounds. Isn't that cool? Well, it's great to see so many people. I want to welcome you to TARC 2024. I think we're in for another wonderful day. Seeing the slides and hearing the talks, comments from last year, I think are a nice prelude to what we're doing today, when we're excited to hear from a number of our junior investigators in our data blitz, and you'll be hearing shortly about what our program is going to look like. Okay. There we go. First, I'd like to thank our TARC Steering Committee. Those of you at member TARC institutions have a steering committee liaison to TARC, and they're represented here in the slide. This is a group that works hard throughout the year to put forth the TARC mission and make decisions about directions for TARC. And it's a very hard-working group that is co-led by myself and Dr. Rodrigo Morales, who you'll hear more from when he'll be serving as our MC today. The latest TARC applications, we received 73 this year. I know there are some of the authors are in the audience, so good luck to all of you. We're really pleased to see so many applications from really representing all of the TARC institutions. And historically, over time, TARC has given out over $21 million to Texas investigators. And again, with funds going to all of the participating TARC institutions since 2018. I'm a little ahead of myself. We're also going to be bringing on board UT Tyler as a new TARC site at the next council meeting, assuming the council approves their inclusion. Okay, my screen's going black, but that's okay. Those aren't. I also want to recognize a couple of our council members that are here. Dr. Sudha Seshadri is here somewhere. Sudha, hold up your hand. Oh, everybody's pointing to you. There you are. Thank you. And Dr. Mark Diamond also is here. Mark's hard to miss because he's so tall. He's here somewhere. Probably getting a banana, but that's okay. I would also like to recognize a kind of a special guest of TARC, since she really created TARC. That's Debbie Hanna is here. Debbie, please raise your hand. She's in the back up right there. Without Ms. Hanna, I like to call her Dr. Hanna, but without Ms. Hanna's work, we've given her official MD and PhDs. But without her work in the beginning, TARC would not exist, and we appreciate her ongoing commitment to seeing that TARC is thriving. So thank you. All of our TARC grants are listed on the website, I encourage you to take a look and see what's there. We fund everything from fellowships and traineeships to large investigator-initiated and collaborative grants as well. The so I love this slide. It says that this is holding pending the announcement. So we had this slide made before the meeting, but we have a couple of large grant initiatives that have been started up over the last round. That includes our Hispanic cohort study as well as our TARC Brain Bank. So we have established a brain bank across the TARC sites. That's led by Dr. Chuck White and who's right back there. And also the Hispanic cohort is led by Dr. John Hart, who's with us. The steering committee recently voted to approve continuation of those projects with some additional input from the committee. So congratulations on that as well. This is all going to be a wonderful continuation of TARC activities. And it's resulted in a lot of extramural or outside the state funding, including NIH, as well as other resources, which was one of the dreams of the TARC Grants program, was to help young investigators get enough funding or some pilot funding so that they could then go on and get NIH and other institutional, other organizational, and other types of funding through foundations as well, which results, if you look at the numbers that we've brought in across our investigator sites, in about a 6 to 7-fold return on investment from TARC. So we think that TARC is doing a very good job. Our investigators are continuing to publish papers and also getting more grants and giving out samples too. So we have a TARC biobank. It's a very active group. And we've sent out over 4,000 samples to people, with more requests coming in all the time. So our database is open, it's available. You just need to contact the TARC office and we'll help you find it and locate it and submit a request for data. So obviously it's been a success so far, and we're excited to hear from everybody that's going to be presenting today. I've got a number, and we're partnering with the Oscar Fisher Lecture Series to provide our speaker at noon. That's really going to be a treat. So hopefully everyone will be here for that. Also, thanks to our staff for arranging the facilities, and to all the workers here. We're going to be back here next year for TARC. We're actually going to have some expanded room upstairs. We're going to go upscale a little bit. It's a legislative year, so next year we're going to want to make sure everybody gets the word spread around even more than this year. Want to see everybody come by the posters this afternoon. I think Dr. Morales is going to give us a kind of an overview of how that's going to work. After lunch and the keynote address, we'll have 3 lectures on different topics running from microbial composition in Alzheimer's, genetic ancestry aspects, which is obviously very, very important for TARC with our Hispanic emphasis in our clinical cohort, and also genetic therapies as well. At 5 o'clock, we'll be announcing the poster winners. So those of you hang out by your posters, be sure you're energetic and talking to people, because you don't know who the judges are. It's secret this year, so that'll be important. And then last but not least, I really do want to give a shout out to our stellar team. I call them our TARC team. It's like my super team they're based. They have multiple duties. They work in my lab. They run my academic life and educational life, and that's been led for a number of years by Dr. Bruce Jones. I don't know if Bruce is actually down here now. Bruce had the unfortunate situation of becoming quite ill as soon as we got here. I don't think it's psychological. We think it was a physical illness. But I want to especially thank Dr. Jones, because actually this will be his last TARC meeting. It's not related to his illness, but he has announced he's going to be retiring in the summer. So we'll definitely miss him and all of his efforts. The good news about Dr. Jones, though, he's also been a great mentor. So he has mentored the rest of our team, including Ms. Nikita G., who's in the back there. And Nikita, raise your hand at least. So Nikita's been in charge of most of this program and figuring things out and getting us good food. So we appreciate that. But she works with a terrific team that includes a number of other individuals, including Alita Godby, who'll be taking over some of Dr. Jones's duties, Sarah Sprinkle. We've got our data managers, Jake Ross Mango and Francisco Argueta, and you'll see our people working the tables out there. So as y'all are requesting data, think about the people you're meeting here. These are the folks you're actually asking for the data from. So that was part of the reason in having them here. We have Ryan Hebinger runs our biospecimen fluids core, and we have Tamika Spears and Ellen are out here working with us, and they're the ones that pull the samples for you. So those of you that are requesting thousands of samples, they're the hard workers behind the scenes. And then we've got a number of student, intern, and postdoctoral volunteers with us here that we're pleased to have as well. This group has worked together tirelessly for many, many months putting this together, and I actually would like to have a round of applause for them in particular. And of course, I also want to thank some of the other people that have really come together and made this event special. That includes our support people and organizers, including Amy Scott at the background, John Da Silva with Roadrunner Pictures, Ben Porter doing photography for us. Ben is an outstanding photographer, and you'll see him running around today. Encore, Elegant Designs, Marquee Event Rentals, The Plant People, Contention Handling Services, and just as you can see, it takes a whole village to put this together. And now I'm going to introduce Dr. Morales and turn over the program to him as we move along. And I think I'm a couple of minutes ahead of time, so take over.
Please welcome the Vice Chair of the TARC Steering Committee, Dr. Rodrigo Morales. Dr. Morales is a professor at the Department of Neurology at the UT Health Houston McGovern Medical School. Dr. Morales has more than 20 years of experience in the field of protein misfolding diseases, specifically in prion and Alzheimer's diseases.
All right. Well, good morning and welcome to this Data Blitz session. Well, as you can see in your program, we have a very busy agenda. So we have 16 talks. So that will be divided into sessions. So each presentation is 8 minutes. So we may have time for questions depending on the length of each talk. But if you have any pressing question like that could take time, we'll have plenty of time for breaks, to discuss And the idea is that you network and you establish perhaps more specific conversations about the different presentations. So, for the presenters, you have 8 minutes. I will be waiting you at 7 minutes, so just for you to wrap up your presentations. And we will start with Dr. Lindsay Goodman from Baylor College of Medicine.
Where's the pointer? Oh, there it is, sorry. Hi, I'm Lindsay Goodman. I am a postdoctoral fellow in Hugo Bellin's lab, and I'm excited to tell you today about my work on tau and glial lipid droplet formation and the resolution of neuronal ROS. So in a healthy brain, there is a nice balance between antioxidant mechanisms, such as glial lipid droplet formation, which I'll go over in a second, and reactive oxygen species, ROS. In aging, there becomes an imbalance between these two features, and this becomes prematurely imbalanced in Alzheimer's disease. We believe this contributes to progression of disease. And in fact, we see that defects in glial lipid droplet formation are likely contributing at this stage. In late-stage disease, it's very clear that there's a high level of ROS in individuals, and this will lead to a further imbalance between these two features. So glial lipid droplet formation is a process by which neurons form peroxidated lipids. So a neuron is the primary source of ROS in the brain. And when high ROS accumulates in these cells, they produce lipids. These lipids become peroxidated and are exported out of the neuron, picked up by apolipoproteins. And the lipidated apolipoproteins are endocytosed by the glia. Within the glia, the peroxidated lipids become integrated into lipid droplets at the ER. And these lipid droplets can be visualized in many tissues, including postmortem AD tissue. Lipid droplets are critical for the breakdown of these peroxidated lipids that originally came from the neurons, meaning we've now reduced the level of ROS in the neurons. And in fact, if we inhibit lipid droplet formation at any point in this pathway, we can cause toxicity in the neurons, glia, or both. We also see that the pathway can be overrun, so if there's too much ROS in the neurons, then the glia actually cannot compensate anymore and they die. So in Alzheimer's disease, there's accumulating evidence that defects in this pathway contributes to disease progression. And here I'm highlighting a number of genes that we've identified that all hit on this pathway and are all important for AD risk as well. Importantly, the AD risk allele ApoE4 is incapable of shuttling these peroxidated lipids between the cells, and as you know, this is a common AD risk gene. So when I started in the lab, I was curious about the disease protein tau and if this could actually induce lipid droplet formation. So in flies, we typically model tauopathies by overexpressing human tau causing in neurons, and this causes neurodegeneration. So when I did that study, I'm not showing those data here, I did not see compelling evidence that pathogenic tau could induce lipid droplet formation. In contrast, I considered whether or not pathogenic tau could play a role in the glia, and I did this because AD patients can have glial pathology, and it's a common pathology in tauopathies such as PSP and CBD. So that's where I found some interesting biology. Sorry. It's supposed to go back. There it goes. So here in the fly brain, I am inducing mild neuronal ROS, and this causes these white puncta to form. These are the lipid droplets. And you can see that when I overexpress human tau, so this is pathogenic tau, we no longer get lipid droplet formation. We can recapitulate these studies from the fly in mammalian systems, including a mouse co-culture model. So here we're overexpressing human tau in the astrocytes, and then we culture them with neurons that carry a fluorescently labeled lipid. When we stress the neurons, those lipids end up into the lipid droplets within the astrocytes. And you can see lipid droplet numbers are severely defective when we overexpress human tau in the system. We also see an accumulation in peroxidated lipids in the system, indicating defects in resolution of ROS. So while I was investigating this from the perspective of pathogenic tau, I was also curious about an endogenous role of tau in this pathway. I was surprised to find that tau is actually highly expressed in the glial cells of the fly, and we now see that it's expressed in astrocytes and oligodendrocytes of mammals as well. And so here, just briefly, we see that in a merged sample, the green is the tau gene expression, the purple is the glial cells, and you can see a large number of the glia express tau. When we lose tau in the fly, we actually see degenerative phenotypes. For example, we see a reduced lifespan when we reduce tau using an RNAi within the glia. So what is tau doing in the glia? I considered, given that pathogenic tau could inhibit lipid droplet formation, whether or not that was indicative of an endogenous role of tau in glial lipid droplet formation. And indeed, that was the case. So here, again, I'm inducing lipid droplet formation by causing mild neuronal ROS. And when we have an RNAi against the endogenous fly tau, we no longer get lipid droplet formation. When we do this in a mouse model, we again see defects in lipid droplet formation when we downregulate the mouse tau gene. And again, we see an accumulation of peroxidated lipids in response to defects in lipid droplet formation. So I don't have time to go over everything that we found in this investigation, but I want to focus on a pathway. So here you see that we have found that both overexpression of human tau, so pathogenic scenarios, that we can cause lipid droplet defects, and we can also see defects when we reduce tau. This correlates with an upregulation in peroxidated lipids. We do see glial cell loss that is age progressive. And again, we see a number of degenerative phenotypes, including reduced survival. I want to really emphasize that the loss of tau is dependent on neuronal ROS. So if we have loss of tau and with no ROS, there are no phenotypes. It's only when there is neuronal ROS that we see phenotypes. Mechanistically, we found that tau is critical for the budding of those lipid droplets off of the ER. And I talked way faster than I expected to.
Actually, we have time for one quick question.
Thank you for that presentation. I was thinking that you have a regarding the lipid circulation, you have a covalent union when they circulate because of the ROS. How can be the drop made because it circulated the lipids, if I am not wrong? Because you have the ROS, the lipid can make cycles because of the bounds that they have in there. So how the drops can be made with this kind of lipids? That's what I'm wondering.
Oh, how does it? So it gets endocytosed. The pathway's not there. The peroxidated lipids get endocytosed, and then there's actually on the endolysosome there are exporters, and there's also direct contacts between endolysosomes in the ER. So there is a transfer. We haven't directly investigated this exact genes, but we believe that there's a transfer of those lipids to the ER. Once it's within the ER, it's well established that that's where the lipid droplets form, and they form with the peroxidated lipids we see that were originally sourced from neurons.
Interesting. We can talk later then. Okay. Thank you so much.
Mm-hmm.
Yeah. Thank you.
Yeah.
Thank you very much.
Sorry for going.
So our next speaker is Dr. Javier Ortiz from UT Health Houston.
Thank you. Good morning. My name is Javier Ortiz. I'm an assistant professor in the Department of Neurology at the Neurocognitive Disorder Center at UT Health. My background is in molecular genetics, but I've been doing clinical science now for 4.5 years. Today, I'm going to briefly introduce an ongoing project whose major focus is to determine whether vaccines lower dementia risk, specifically Alzheimer's, in older populations across the US. Now, to tackle this question, we actually utilize team science, so we have collaborations with statisticians, experts in bioinformatics, clinical and translational scientists. And we think that this is a worthy question because it's more beneficial to investigate interventions that prevent the disease rather than to deal with the disease. So there are previous studies that suggest that vaccination reduces the risk of developing dementia. However, these studies are quite limited, either because they focus on small cohorts or on specific populations. So what we would like to do is we'd like to ask this question: in a way that it reflects the entire US population, but at the same time only focuses on Alzheimer's disease. So to do this, we employed a large database called Optium Informatics Data Mart. And this is health insurance claims for more than 67 million Americans across all 50 states. And this insurance claims include medical claims, pharmacy claims, lab results, and information whether a subject attended specialty clinics. To generate and evaluate our cohort, we use a 10-year time frame, and that includes the first 6 years we used to generate the cohort, and the last 4 years we used to evaluate the cohort. And for the criteria for a cohort was that subjects had to be 65 years or older, that they had to be dementia-free, that they had to have status of vaccination, and once we generated the cohort, we used an algorithm to synchronize the baseline variables and demographics of those groups. So at the end of the day, we basically had subjects that had no vaccination or at least one vaccination. And we then evaluated this cohort using relative risk to assess vaccines' effect in the last 4 years of that 10-year time frame that I just mentioned. So first, we focused on influenza vaccine. So we first generated our cohort, and then we applied the algorithm that I just mentioned, and we produced 2 groups of vaccinated, of no vaccine versus at least one vaccine, and each group had 935,887 subjects. And what the results show is that more than 47,000 people who had one or more vaccines developed Alzheimer's disease during this 4-year period. However, if they had no vaccine, that number almost doubled. So the results show that adults ages 65 and older were 40% less likely to develop Alzheimer's disease compared to the unvaccinated group. So then we ask, is this result specific to the influenza vaccine, or is it a general property of vaccination? So then it turned off. Sorry. Okay. So then sorry, it's just keep on going back and forth over here. Oh, so then we ask whether this was specific to one vaccine or multiple vaccines. So then what we did, we investigated whether vaccinations in either, whether specifically in subjects that had 6 vaccinations, 3 vaccinations, or no vaccinations. So what this result shows here is that subjects that had 6 vaccinations have less incidence of Alzheimer's disease compared to subjects that have 3 vaccinations or no vaccinations. So then we tested this subject, or this same principle, because we wanted to test whether this was specific to influenza or was a general property of vaccination. And we tested it on 3 different vaccines. We used the same retrospective cohort and the same algorithm. And we tested it on tetanus and diphtheria, on herpes zoster, or pneumococcal vaccines. And then what the results show is that vaccinated groups were 30% less likely to develop Alzheimer's disease compared to non-vaccinated groups in the tetanus and diphtheria. 25% was for the shingles or the herpes zoster,